Bismillahirrahmanirrahim. Facilitated diffusion. Before discussing the facilitated diffusion, remind the classification of mechanism of absorption is passive absorption, active absorption. Passive is divided in simple diffusion. Simple is then convective transport, acid-base diffusion, ion pair. Facilitated diffusion is channel mediated, carrier mediated, and filtration. After these all are the mechanism for passive absorption. Now, in passive absorption, today we are discussing facilitated diffusion. Facilitated diffusion is of two types. One is channel mediated diffusion and one is carrier mediated diffusion. Let's discuss the carrier mediated diffusion. This carrier mediated diffusion is sometimes considered just like active transport, but the difference between active transport and this facilitated diffusion is that facilitated diffusion don't proceed against concentration gradient, while active transport proceed against the concentration gradient. दोनों में फ़र्क ये है कि एक्टिव ट्रांसपोर्ट हमेशा अगेंस्ट कंसंट्रेशन ग्रेडियंट होता है विद द हेल्प ऑफ एनर्जी, जबकि कैरियर मीडिएटेड या फैसिलिटेटेड मीडिएटेड ट्रांसपोर्टेशन ये अलोंग द कंसंट्रेशन ग्रेडियंट होता है. तो इन दिस कैरियर मीडिएटेड ट्रांसपोर्ट देयर आर सम इंट्रिंसिक कैरियर just like intrinsic factor which are produced by the stomach wall. These intrinsic factors form complex with vitamin B12. This complex is then internalized into the cell, that is this complex crossing this biological membrane, and now inside the cell the lysosome cutting this complex, and now after cutting the B12 is free and this carrier, that is intrinsic factor, is free. Now this intrinsic factor is now again reabsorbed to carry next vitamin B12. By this way about 1.5 microgram vitamin B12 is absorbed per day by this mechanism. Other drug example including cephalexin, captopril, levodopa, etc. The second type of facilitated diffusion is channel mediated. In this case, special type of channel protein, that is facilitator proteins, FP proteins, are involved at the cell surface. Once the ion channel drug etc. form complex with these channel, before the complexation these channels are in closed form, but once the drug bind here, just like in substrate enzyme form, specific shaped drug bind with this specific binding site, this complexation inducing the opening of the channel. Once the channel is open, the drug is internalized and go into the cell, that is absorbed, crossing the cell membrane. Or sometime this drug form complex with the special protein at the cell surface. This complexation changing the this complexation of drug and this channel, inducing the conformational changes. Now look here, this is the channel, drug, biological membrane on this side. Once the complex is formed, there is conformational change and twisting. Look here, this is the drug and this is the channel. Once the drug channel complex is formed, there is conformational changes, and now now the drug is on this side of biological membrane, and the protein is on this side. This is known as conformational changes. So the conformational changes. After the conformational changes, the drug is released on the other side of biological membrane, and the protein restore its shape. This mechanism is also possible. And other drugs acting through this mechanism are sugar, amino acid, nucleotides. The last thing is this this lecture is filtration. Filtration is the penetration of drug, ions, molecules, whatever, in the pores of biological membrane, just like in glomerular filtration. the drug penetrating in the pores through the osmotic pressure. This is very important. This penetration is very important in case of glomerular filtration. But this is also important in case of intramuscular injection. Once a person injecting intramuscular, the drug accumulates in the muscle. If we rub that area, there is vasodilation. After vasodilation, the opening or the diameter of the pore is increased and the drug is internalizing. Thank you for listening.